Hello, I'm Jerry Spivak. I'm an emeritus professor of medicine at the Johns Hopkins University School of Medicine, and I'm going to discuss today how research developments in polycythemia vera can provide insights into acute myelogenous leukemia. Now, it seems obvious when I make this statement that these are two apparently different disorders, but their differences are as important as their similarities. Now, both disorders arise in a pluripotent hematopoietic stem cell, except for acute promyelocytic leukemia. And both disorders involve the overproduction of blood cells. In polycythemia vera, obviously, the cells are phenotypically and functionally normal. And in acute myelogenous leukemia, they are inert blast cells unable to differentiate. Now, the other important difference, or one other important difference between these two disorders is that left to supportive treatment alone, and that means no chemotherapy, polycythemia vera patients have lifespans that can be measured in decades and often are normal. However, in acute leukemia, without chemotherapy, the lifespan is measured in months. Now, what this means is that when studying acute leukemia, one is faced with the end product, the blast cell, which is already genetically severely damaged. While in polycythemia vera, one has a stem cell that has one mutation in JAK2, and the stem cells are otherwise apparently normal. So one can follow the evolution of the disease, and polycythemia vera patients get all three types of acute leukemia, namely the de novo type, the secondary type, and treatment-related acute leukemia. And it turns out that as polycythemia vera patients develop myelofibrosis, they tend to have a tendency to transform to acute leukemia, so that one can follow changes in stem cells over time and understand the mechanism by which leukemia occurs, because the ultimate leukemic mutations occur in polycythemia vera, just like they do in acute myelogenous leukemia. Now, in addition, most patients who develop acute leukemia are over the age of 60. Most patients who develop polycythemia vera are over the age of 60, and most of those are men. And men more commonly get acute leukemia with polycythemia vera than women do. So these are important differences and important similarities. Now, what happens over age 60 is that as we age, we unfortunately tend to develop lesions in genes that cause clonal hematopoiesis. And two of the most common are TET2 and DNMT3A. These are sort of fellow travelers. And we call this CHIP, clonal hematopoiesis of indeterminate potential. I rather prefer the phrase ARCH, age-related clonal hematopoiesis, because it pinpoints where it starts. And we know that in a hematologic malignancy, CHIP can be a serious complication, which has definite known potentials. JAK2 is a unique mutation because it allows cells to pass through G2 with great efficiency, or go through the cell cycle. It's a better way of saying this, with greater efficiency. And if there is another mutation in the cell that damages DNA, normally the gene TP53 will block that cell from going through the cell cycle. But JAK2 can overcome that block. And unfortunately, one of the genes involved in CHIP or ARCH is TP53. So if you have a mutation in TP53, JAK2 can overcome that mutation and allow a damaged cell to suddenly be able to outcompete normal stem cells. Now, it happens that people get diseases like polycythemia vera because they have a genetic background for same. And this usually rarely occurs in families, but it's more commonly due to germline SNPs, or single nucleotide polymorphisms, that are inherited. And the JAK2 SNP is the one that causes increased risk for polycythemia vera. But recently, another SNP has been associated with the myeloproliferative disorders and polycythemia vera. And that is one in a gene called HMGA, high mobility group protein A, which is a chromatin modifying protein, opening up the chromatin to allow transcription of genes that otherwise would not be transcribed. This gene is a definite oncogene causing solid tumors and acute leukemia. Now, we've been studying polycythemia vera transformation to acute leukemia, and we found in a transgenic mouse model of polycythemia vera that the expression of HGMA 1 and 2 are elevated as the mice transform into the myelofibrotic stage of polycythemia vera. And we found that in human polycythemia vera stem cells, the HMGA genes are also increased in expression as the transformation occurs in humans as well as in the mice. Now, we have a mouse model of polycythemia vera in which we can induce acute leukemia. This is a fulminant acute leukemia, which primarily involves megakaryocytes and erythroid cells, and is very much like the leukemia that you see with TP53 mutations. And we have found in this leukemia, by silencing the expression of HMGA1, we can increase the survival of these mice almost triple what would occur in the untreated leukemic mouse. So this provides a target not only for treating acute leukemia as it occurs in polycythemia vera, which at this point, no treatment has been satisfactory, but also a new target in patients with acute myelogenous leukemia. And we know that downstream of HMGA is GATA2. So these appear to be GATA2 driven leukemias. Now, we also know that if you give chemotherapy to patients with polycythemia vera, you can cause acute leukemia. This is true for any chemotherapeutic agent, but unfortunately, the most widely used acute leukemia chemotherapy in polycythemia vera is hydroxyurea. And hydroxyurea has several deleterious effects. One, it can cause a P53 mutation on its own. Two, it can inactivate TP53. And if you have a JAK2 mutation and you have hydroxyurea inactivating TP53, then you've essentially thrown gasoline, a match into gasoline. And it's been shown that in patients with polycythemia vera, chemotherapy with hydroxyurea increases the incidence of acute leukemia. And finally, we think of polycythemia vera as a disease driven by the JAK2 mutations. This is true certainly phenotypically, but JAK2 also on its own, as I mentioned, inactivates P53 by upregulating several other genes, such as MDN2 and MDN4. And these genes express proteins that also block TP53. Now, as we've found in a mouse model of Polycythemia vera, that these cells, that the stem cells are actually sensitive to thrombopoietin. And it is well established that thrombopoietin levels are elevated in the myeloproliferative disorders. And in a transgenic mouse model of Polycythemia vera, we've been able to change the phenotype to normal by knocking out the thrombopoietin gene. So this is a second mechanism to deal with a disease that's prone to acute leukemia. And it's also been found that by knocking down thrombopoietin, that there's been an improvement in mouse models of breast cancer. So this remains to be looked at carefully in acute leukemia, but stem cells have only one growth factor receptor, and that is the thrombopoietin receptor. So this remains a potential target of opportunity in both of Polycythemia vera and acute leukemia.